Welcome to Bio Practical class. Today we will do the study the action of salivary amylase enzyme on starch. Salivary amylase enzyme which can digest starch into maltose, isomaltose, dextrine inside our mouth. Now this experiment is we will take out a saliva and we will do this experiment outside the body in test tube. So this is quite amazing experiment. This is tyline, which is salivary amylase, another name, and here the starch with the help of salivary amylase, it splits into starch is a polysaccharide, and now it breaks into maltose, isomaltose, disaccharide, and monosaccharides. Okay. So let's come here in the experiments. In experiments, so many material required you can see it here, but at first we have to know about the starch, about the sodium chloride solution, and the saliva. These three items that we have to prepare. Let's know that how we are preparing it. First we have to take starch, 1 gram of starch we have to wait, and we have to put inside the 100 ml of water, so that we can make 1% of starch solution. Same way that salt also 1 gram salt we have to take, and we have to mix it in 100 ml of water. In that way 1% of sodium chloride NaCl solution we can make. So starch and the sodium chloride, it is prepared in this way. Okay, starch will take the starch and will apply the salivary amylase. Now I will tell you that, and after that it will digest into maltose, isomaltose, dextrine. But how we can understand that it is broken into simple carbohydrate. So here is that iodine test that you know. Iodine test can indicate that. Suppose if starch, if we apply the iodine, here it shows that the blue black color. After applying salivary amylase and in time duration, 2 minutes to 10 minutes, gradually we can see that this blue black color, when we are applying that iodine in different time level, when the iodine will, after applying iodine, we will not get any color, any blue black color. So we can understand that the starch now, it is not starch, it is now converts into maltose or glucose. Okay. In iodine, this type of breaking will be there. Here you can see this one. Here, so this is starch. So this is starch. Here a branching structure is there. After action of the salivary amylase, here it splits into maltose, the disaccharides, and after that monosaccharide, that is glucose, that limit dextrine, which also is much simpler than this. Now in iodine, it shows the color in two different that we have to know little bit. Look at here, the starch contains starch here, it contains the two polysaccharides, one is amylose, another is amylopectin, Amylopectin is branching structure is there, and amylose also the long chain is there. So in this long chain, look at here, in longer helical, when it is there, that triiodide, this iodine triiodide, it trapped over there. See that the chain, and so it shows one certain color, that is the blue, purple, black, that kind of color. But when this chain will be broken, it means that it goes in simple limit dextrine or maltose or glucose, so that iodine will be not trapped and there will be no color, only iodine color, the yellowish color will be there. That is the reason that in starch it shows the blue black color. All right. Normally the setup here, it is like that. Setup is that experimental tube, which is this one. Experimental tube, we are taking the starch 5 ml 1%, sodium chloride 1% 1 ml, and the saliva, it is diluted 1:20, that we are taking, and we are keeping in water bath which is 37 degrees Celsius. In that we are keeping the temperature, because inside our mouth, inside our body, that 37 degrees Celsius is there. We are keeping all the environment which is there. Sodium chloride, it acts as an activator here for the digestion, which is there in our saliva. That is we are applying here, and the saliva also it is taken out from our mouth, and starch solution. In that way, in first first minute, if we see that the minute wise calculation is, the first minute it will be blue black color, because it will be starch. After 2 minutes also we can get the blue black color, because the digestion process just started, so it is not broken into the glucose. After 4 minutes, that we can get the intensity will decrease of the color. Intensity will decrease means that the long chain, when it will breaks into the shorter chain, and so the color will also decrease. And after 2 minutes, it is the intensity will decrease. Next one, blue black color faded or disappears. And after 10 minutes, if we can see that there is no blue black color, it means that the whole starch material, it converts into maltose, dextrine or glucose, so that there is no starch material is there. Here we are doing the experiment. First we are collecting that saliva. Look at here, that from the mouth. After that we are making it dilute into 1:20, means 1 ml saliva if it is collected, then 20 part we are adding the water, so the saliva is diluted. Actually in our mouth also it works like that. Then we are making ready for the water bath. Here we have to keep it in, at first we are taking it up to 40 degree, then it will gradually decrease into 37 degree. Here is the starch, 1% starch and 1% salt solution is there. Then first we are taking 5 ml 1% starch solution in a test tube. Here it is. This is the 5 ml it is taken. Then after that the 1% initial sodium chloride solution, it is added in that same tube. So we are creating the environment of mouth, and this is the dilute 1:20 dilution is there, that saliva, only 1 ml of saliva that dilute saliva we are taking, and we are mixing it, and immediately the digestion process will start. So, we have to transfer it in water bath. Water bath, already it is 40 degrees Celsius. Now it is a little bit reduced, so it is 37 degree, and we have to keep it 37 degree. After keeping after 2 minutes we are taking it out, and look at here, it is the dilute iodine solution we have given in the every part here. The first, after 2 minutes when it is taken, then it is quite dark color. Then after 5 minutes it is taken, then it is you can see this is the digestion after 7 minutes, and after 10 minutes. After 10 minutes it is uh, okay. Here this is the first one when it is I mean after 2 minutes. Then after 5 minutes when it is given, you can see that the color difference here. Gradually it converts into maltose and glucose. Then after 7 minutes you can get the lighter, and the after 10 minutes when it is there, no black color is there. After dropping the same amount, same drops in every square of it, and here we are doing in 37 degree Celsius. This practical can be done in the 5 degree and the 70 degree, that also in your syllabus. So, that is the way that we have done. Thanks for Rahul to help me this all the setup. You have to prepare this part. Okay, thank you.